Hello, my dear O level students. In our last videos of microorganisms and biotechnology, we were talking about viruses, we were talking about bacteria, and here in this video, we are going to talk about fermentation. We need to talk about the design of fermenter, and we will also talk about the ethical concerns of bacteria and why bacteria is preferred to be used in genetically modified products. So, here on your screen, the objectives ask us to discuss why bacteria are useful in biotechnology and genetic modification. For this, let me give you a little example. Consider we need to make insulin. For example, there is an inquiry in which you may need to make insulin. So, what you do is you extract DNA from from the pancreas. This is your pancreas, and this is a gene for insulin production. Now, cutting a long story short, what we do is we take a bacteria